looking toward the horizon of risk assessment, and the talks that we put together are going to provide a variety of perspectives around this transition that we've talked about. And starting us off, Lauren Zeise from the California Environmental Protection Agency is going to talk a little bit about historical perspectives of risk assessment and how that's affecting some of our thinking about the way we make this transition. Lauren?
This one. Okay, well, the first thing I want to do is give a disclaimer that I'm not speaking for the state of California or any institutions within it. So what I'm going to do is basically talk about two things, and I very purposely put the Little Red Schoolhouse on this slide because especially after Kim's beautiful talk, this is going to be a very old school look at what have we learned from the Redbook forward. And so I'm going to talk some about the Redbook approach and also some lessons learned as we implemented that approach going forward in terms of making inferences, in terms of making decisions, and considering decision contexts and themes. So pre-Redbook, what was going on? Well, let's see now. EPA had been in existence for about 10 years when that committee started meeting. We had a lot of horror stories in terms of environmental exposures and a lot of statutes, new statutes to do something about them. At the same time, the National Toxicology Program was presenting or developing a lot of new testing information on the heels of a National Cancer Institute program. And the two programs together were kicking up a lot of information on chemicals that were out there in the environment. And there are many different ways of interpreting what those data meant. So there were different camps that were looking at the hazard signals saying, well, there's no hazard here at low levels. There were a variety of dose response information. It was pretty contentious in terms of moving forward on toxic chemical regulation. So enter the Redbook. And the Redbook served to begin to bring some organization and rules and ways of thinking about the kind of information that people were trying to make decisions on. So the book was very general. It wasn't just developed for EPA, but across the different federal agencies. And it put forward a framework for thinking about how we look at risks and how we look at management. And this is the 1983 Redbook Framework. So you see that it starts, the thinking was we're going to start with research. Research then begin to surface problems, and the problems are dealt with by considering hazard identification, dose response, the exposure assessment. These different analytical components then feed into a risk characterization. This risk characterization feeds into the decision, so a very linear process. So one of the things the committee, this early committee, was very aware of was the difficulty in moving ahead and making decisions. And they chalked it up to two things. One is basic inherent uncertainties in the analysis, and two were practical constraints of the external world. And that's why they had this separation between risk assessment and risk management. It also noted that uncertainty is pervasive and that conclusive direct evidence of a threat to human health is very rare, and that risk assessors must rely on a series of assumptions. So what this committee recommended was that federal agencies develop uniform inference guidance to use in risk assessment. And an inference guideline is an explicit statement of choice among all the many options that arise in inferring human risk from data not fully adequate or not drawn from human experience, so from indirect evidence. And the kind of evidence that we're going to be facing in terms of from a systems biology perspective, there will be very many different ways probably of looking at the evidence. And there may be a lot of contention about those interpretations. So the Redbook Committee had in front of it a way of thinking about evidence of hazard, where you had endpoints from human epidemiology and toxicology, and that provided direct evidence of carcinogenicity, but they also recognized less direct evidence, where you might have mutagenicity information and you might have structure activity information, and these feed into the assessment. But that committee decided that, and it gave some guidance on where it thought the federal agencies should come out in terms of structure activity and these in vitro assays, and that it really focused on direct evidence from in vivo and human studies. And we're pretty much there today to a certain extent still. So just wanted to point out that the agencies, and I'm going to focus on EPA, after this recommendation for inference guidelines, proceeded apace to develop guidelines in the early part. And so by 1986, we had guidelines for carcinogen risk assessment, then we had guidelines for neurotoxicity assessment, guidelines for developmental toxicity, othericity, we have reproductive toxicity guidelines, and then on specific circumstances, like how do we interpret information on thyroid tumors in animals. And then for place-based assessments, how do we go about analyzing information on exposure and dose response in terms of making decisions about Superfund sites. And most recently, the agency, I think this is 2010, updated its guidelines for how we go about estimating toxicity information for dioxins. And then in 2005, on how we look at early life exposures. So there's this whole body of information on inference guidelines that guide how the agency makes decisions about toxicity. And some of these guidelines didn't come about very easily. So what are we going to do now? We're kind of looking at, in the near term, potentially making some decisions about systems biology information and integrating it into our assessment. Should this be a very slow walk? Should we take the dive and move more quickly? At what speed should we go as this information's emerging? And as the larger paradigm about what this grander toxicity testing system might look like. How might we move towards making decisions with some of the information that's newly emerging? So Kim already has done a beautiful job describing the toxicity testing in the 21st century, so I just want to go through and just mention some of the components. Again, we're talking about information, high throughput information on non-apical endpoints through high throughput tests like ToxCast, but also some medium throughput assays of more integrated cellular responses. We're talking about toxicity testing in whole animals and maybe even some clinical studies in humans, kind of guiding our path on areas where we don't have the same degree of confidence in making predictions, but in the nearer term, we will have that kind of information regardless as we implement and continue to explore different toxicities. And then around the outer edge is this whole issue of population data, surveillance data, but also, and we're going to be hearing more about it today, there's a lot of information coming from metabolomics and different ways of probing the human condition to see what kind of health status and where are we with respect to some of the pathway data that will be coming out as we move forward in this paradigm. And then, of course, there's the risk concept. So this was a very important component to kind of think about in terms of data interpretation. So how do we begin to make inferences? Well, The EPA already, and it has a system, as we pointed out with the dioxins, for looking at upstream information for making some decisions, and it's beginning to incorporate some of the systems biology information into its thinking. And the thinking follows along this simplified source to disease paradigm, very linear process, where one moves from dose to from an environmental exposure to an environmental transformation to a dose to a precursor effect. And these guidelines in 2005 lay out ways of thinking about mode of action and how to use in a regulatory context, in terms of a risk assessment for regulatory action, these precursor effects. So this information in this approach of this guidelines approach to addressing the biology is laid out in these guidelines. Now these were again in 2005, and they came after the 1986 guidelines, and there was a whole lot of biology being done in between 1986 and 2005. So these guidelines were a long time in coming. There were interim guidelines that the agency used along the way, but this was a very contentious process to come up with these guidelines. So the question is what to do? How do we move this forward? And one of the approaches that the agency took was it basically looked at the way in which it was using its defaults and decided that, well, we're going to look at the data first. We're going to look at all the data first before we make a decision. Now, the problem with that is that led to a lot of arguments. The agency really got bogged down. So some assessments took 10, some even 20 years. We still are waiting for a dioxin. We have the dioxin. We're waiting for a piece of the dioxin assessment. But some assessments, the point is that some assessments take a very long time. And when the Science and Decision Committee looked at this whole approach, it really called into question whether or not this ad hoc, complete reevaluation of the information as you try to move ahead is the best way of going. And it called for maintaining defaults, but maintaining the system of defaults in a way where you could develop much more rapidly a practice in an inference system without with a much more expeditiously. So that was clearly I think a lesson learned for our whole activity here, where now in the near term we're thinking about hazard traits having both direct evidence from apical endpoint data, but in some cases that's going to go away, and all that we're going to have is other relevant data. So what kind of an inference structure can we develop around some of the emerging tools and some of the structure activity and other information we already have in front of us, so that we're much more rapid in our development of assessments that can serve risk decision processes. Another key issue around, and we've already discussed this some, is we may not necessarily know specific cancer endpoints that will be would be implicated by certain activities, and we won't necessarily know reproductive toxicity endpoints, so there'll be lack of definition at the endpoint level. Does it make sense to try to discuss this in a probabilistic fashion? What should our approach be? So this just points out that, so what I want to do here is just point out that there is currently a varied set of data facing different agencies and institutions that are trying to make decisions about hazard. So if there's sufficient evidence in humans, obviously there's sufficient evidence. The chemical can be labeled as carcinogenic to humans. But what about if all you have is convincing evidence from other relevant data, for example, from in vitro evidence, from non-cancer bioassay evidence? So currently, IARC and NTP have flexible enough guidelines to begin to address it, but what isn't covered with the emerging evidence is a system of practice. So within that blue box, to a certain extent, there's general guidance, but a lot of it is ad hoc. And I think for regulatory agencies, as they try to move ahead, that's going to be very difficult. And we will note that there are very few cases where, in fact, a chemical has been identified as a cancer hazard based on inadequate evidence from an apical endpoint view. So this, again, it will be a very difficult as we try to move ahead to incorporate some of the newer data. So making decisions, one thing that the Redbook did was they separated risk assessment from risk management. And so there was this, in a way, it's kind of a handoff. The risk assessor does its work, it hands off the works to the risk manager. And what happens is that for a variety of decisions, the kind of information from the risk assessor might not, maybe the risk assessment took too long and they weren't able to produce the information for the assessment, or maybe the information was of a different nature than needed to finalize it. So the NRC committee in 2009, there were a variety of committees along the way that had looked at this whole process and called for much more engagement at an earlier stage in the process by stakeholders in thinking and formulating what kind of information was needed to make a decision. And this risk assessment committee that produced the Silver Book focused on changing the process early on in terms of designing the risk assessment process to think about what kind of options are in front of the managers for answering the question of, for which the risk assessment is designed to answer the questions, and to put a lot more effort earlier in the design process. So, and to have the, so just point out, as part of this process, the standard 4 steps were part of this. So again, in terms of the toxicity testing in the 21st century paradigm, risk contexts frame the outer circle and are part of thinking about what kind of systems we need. And I think it's probably plural systems in terms of toxicity testing. What kind of systems we might need to answer some of those risk assessment questions and decision questions. So the issue is, how do we design the risk assessment systems to match the needs of the risk manager or risk management systems? We kind of need the right level of analysis and the right level of testing, and to have a good fit. So in some cases we might be able to move forward very quickly. In some cases we actually might need something like a good understanding of low-level risks. So I just want to run through quickly some of the kinds of decisions that some of the kinds of decisions in front of the committee and in front of the agencies. And one of them is this whole issue of no traditional test data, no risk. So that's what's going on right now. In the near term, can we move away from that, and how quickly? Right now, in vivo evidence leads to a risk assessment, leads to a decision, leads to regulatory action. So again, that's out of science and decision, is a call for looking in a probabilistic way at structure activity and in vitro evidence to come up with a probabilistic description of how risky a chemical may be. And Ivan Rusin and Kate Guyton and others at EPA are beginning to design a system based on what we know about in vivo evidence of toxicity and in vivo measures of toxicity, to integrate this evidence and come up with a prediction of how toxic a particular chemical is. So it's trying to move, and I'm been part of this process as well. We're trying to move forward and begin to think about some of the emerging evidence and how we might use it to predict toxicity values in the nearer term as the system is being built and as we're learning about biology. Another issue is in terms of screening and prioritization. We are going to have in front of us, as we begin to think about some of the green chemistry initiatives, and some of the bad actors are being identified by in vivo evidence, but then the question is, what do you substitute it with? And can the information that's being developed now and in the nearer term help make some informed decisions about what we replace it with? So that's a big issue. And then if we're thinking about, well, what spectrum of exposures might be at different sites or different facilities that we might not have test information on? Can we use some of this early information to prioritize which chemicals that might be out in the environment that we test, where we might put our monitors, where we might put our enforcement activities? So screening and prioritization can be very important. What we've learned out of the various evaluations of EPA work is that cumulative risks are an important consideration. So here is an example from the phthalate report where the committee analyzed information on chemicals that operated and affected the status of androgen receptor activity. And they had operated by different modes of action, and they affected that. They disrupted androgen processes by different mechanisms. And when you looked at individual studies, you saw that for an individual chemical, it could be tested below a threshold and pose no risk. You test another chemical, no risk, operating via different mechanisms. But then when the chemicals were tested as a mixture, you had a response. So the the committee picked through these data and came up with an overall paradigm for looking at cumulative assessment. And so it was pretty clear, at least for the chemicals that committee looked at, that going single chemical at a time wasn't really getting you the right answer. So the question is, from a systems biology point of view, how might we look at that data and how might we design our systems so that we capture this problem? So I just want to point out that this emerging sciences group had a workshop last year about this issue. And then what about non-chemical stressors? So if we look across from an epidemiological point of view, what do we see? And this is a study that Dan Kruski et al. Published on the left that shows mortality versus high school attainment for different air pollutants, sulfates, and fine particles. And you see that high school attainment was an important factor. There are various other studies you're seeing that you see associations for traffic-related pollutants, low birth weight, and low socioeconomic status compared to high socioeconomic status or average socioeconomic status, with effects in the lower group having much greater effect. Exposure to violence, parent stress level. So these non-chemical stressors turn out from an observational point of view to be important. How can we look at this from a systems biology perspective? How do we think about our interaction of chemicals with non-chemical stressors? And again, I think today we're going to hear some information on this point that will be very helpful. And then the Science and Decision Committee looked at this dichotomy to the way we analyze dose-response information, where if you look at an individual chemical, you actually can see some fairly steep dose-response relationships in very homogeneous animal tests, where you have all animals very similar. And Kim showed a beautiful example for bisphenol A, where with one set of background conditions, you get one set of responses, and another set of background conditions, you get a different response. It looks safe or even protective in one case and not protective in another. Well, maybe the human population is made up of people at all kinds of different status. So the question is, what can we say about background? And in addition to what we're exposed to from our environments, we all come at this from genetically from a different, and so our endogenous exposures differ across the population, sometimes quite dramatically. And there's the epigenetic issue as well. So when we think about all of these things together, individual responses are going to differ. How can our test systems, what we create, take into account, and how can we use systems biology to get at what might be important risks at the population level? So what the Science and Decision Committee thought about was in terms of apical endpoint data, in terms of our epidemiology, although we were looking to upstream kind of evidence for interpreting the evidence and thinking about how we combine and how we analyze and come up with an estimate of population dose response. But a critical factor is thinking about background and how it influences the exposure of the chemicals that you're recommending as safe or not safe. Or perhaps we need a more probabilistic approach. So again, that's science and decision. So I'm really looking forward to the discussion at this workshop, and I think that systems biology has perhaps some potential in the nearer term as well to help inform some of our decision processes in matching and getting the right kind of risk information to the particular decision at hand. Thank you.